Hi. Hi guys, I'm live on the YouTube channel if you want to come over and join me there. You'll be able to see my resources. And if you actually want to join the live, then the Zoom session, the link is in the chat. If you would like to join me, I am on the YouTube channel with my screen. And if you click on the participation, I will be going through some questions and topic areas. So hi guys, if you're just joining me, if you want to jump over to my YouTube channel, I'm currently on there with my screen share, so you'll be able to see the content I'll be covering. Hi Chidima, sorry, I almost said it wrong again. So I'm over on YouTube as usual, if you want to come and join me. And guys, if you want to actually join the session, then do just click on the button. Is there any specific topic areas people want to do? Otherwise, I will just start with a question paper and work our way through it. Okay, so what I'll do then is I will start the session looking at some questions. So I'm going to look at a paper 2 today. So I've done some paper 3 work. So I've got 2 paper 2s that we can work through. Do come and join me if you would like to actually go through some questions. So I'm going to start with looking at, so you can see here, I have got, make it a bit bigger, should hopefully, oh, a bit too big. Make it a bit bigger for you guys on the screen. And if you want to join them, please do click on the link. There we go. Okay, so if we look at this first paper then, and we work our way through how to answer these questions. So if you're just joining me and you want to come over and look at the paper that we're currently looking at, we are doing a 2021 paper 2. So we're going to start with 2021 paper 2. So if you're interested in the paper, and we will start with our multiple choice, and then we'll work our way through from there. So the best thing you can do whenever you're doing practice papers is to try and identify how they got the answers wrong, which ones are the wrong answers, and then use that to guide you on which ones are the right answers. So if we then start with this one, which of the options is a function of fibrous protein. So this was one that we were covering last week. Okay, so you're saying it can't be D because enzymes are globular proteins. So this is an interesting one because you're talking about speeding up the rate of reaction. So the rate of reaction we know would be the enzymes, because that's what biological catalysts are. They are enzymes that are going to speed up the rate of reaction, and you know that enzymes should be globular proteins. So good, we are confirming that knowledge there. So then what we want to do is we want to ensure that that then leads us with our right answer. So the next thing you said is not B, because glycoproteins are let me read that again, are responsible and they are conjugated. So provides elasticity in alveoli involved in cell signaling or aids rigidity of membranes. So you said it's not B because proteins are responsible. I see what you're saying now. So you're saying that this is your glycoproteins, and the glycoproteins are involved in your cell signaling, and they are as well globular proteins. So they are rounded in shape, so it's not our B. And then you have said that it is as well, rigidity of membranes is cholesterol. Yes, that's right. Rigidity of membranes is cholesterol, so it cannot be a cholesterol. It's not even a protein, it's a lipid. So rigidity of the membranes, as you have rightly said, is cholesterol. You're on fire tonight. Well done. So therefore, we can get rid of A, leaving us with the only right answer. You're welcome, which is C. So which of these is a fibrous protein? Now, just to remind ourselves, what were the 3 that we needed to know for last week's lesson? So last week when we did this, we talked about the 3 that you need to know, which are your collagen, which is your keratin, and is your elastin. So therefore, we are obviously annotating. Whenever you do these practice questions, always annotate with the extra bits of information, because that's going to help you in how to be more successful in answering this. Right. So carrying on then, let's move on to the next question. So the minute we get a question like this and it tells us we've got a type of molecule, I always say, right, we've got this type of molecule. What can I see? I would identify some key parts to it. Let's try that again. So that I can see, well, straight away, I can see over here, we have a nitrogen containing molecule. So because it's a nitrogen containing molecule, I know that this has got an amine group. So we've got an amine group here. Then we also have on the other side a carboxylic acid group. Oh my word. Let's try that again. Carboxylic acid group. Or carboxyl group, if you want to shorten that down. And then we have our last little section here, which is our R group. So we've got the different parts to it. So let's remember, it's about how we eliminate the wrong answers, not just about confirming those right answers. So it is really important when you're doing this that you actually work out why the wrong answers are wrong. So we know the minute it's got an amine group, we can get rid of our fatty acid straight away. Fatty acid would be only a carboxyl group and a long chain of our carbons and hydrogens. The other thing that we can note is a nitrogenous base. Well, if we're talking about nitrogenous base, we need to think about whether it's a purine or whether it's a pyrimidine. Now, remember, when I go through this, pyrimidine, I'm never just going to give you the answers. That's not the point in these sessions. The point in these sessions is to help you revise around the whole topic area to ensure that you are getting the most out of this. So whenever you're doing the revision, don't just do multiple choice in a way where you are guessing the right answer or just stating that that's the right answer, because what they can do is they can take a question like this, a multiple choice question, and remove those A, B, C, D options and now turn this into a 1 marker, a 2 marker, a 3 marker, and ask you, how could you identify which molecule it is that you're looking at? So it's important that you think around all the topic area. Okay, so we know that there's not a purine or a pyrimidine because we know that the purine has got a double ring and a pyrimidine has got the single ring, and there's no ring in this diagram. So we know that it's not a nitrogenous base. A nucleotide, we know it's not a nucleotide because the nucleotide would have to also have the phosphate. There's no phosphate there. Also, it doesn't have a pentose sugar. Now, I've said it's a pentose sugar. I haven't said it's a deoxyribose sugar. I've said a pentose sugar. And the reason I've said pentose is because you should know that the nucleotides could be DNA with a deoxyribose sugar, or it could actually be RNA with your ribose. So they haven't shown that, obviously it's not in the picture, but in the picture it would be something you'd be looking at. What have they put at carbon number 2? So if you can identify that, that will help you so you know how to answer the question. So for those of you who are joining me on TikTok, I am currently live on my YouTube channel, and that is showing you the screen with all the questions. So if you want to jump over, please do feel free to come and join us over here, and I will continue with the questions. So the next question then, which of the following processes is important in determining the overall body plan of an organism? So this one, because it's looking at the body plan, it's looking for your content knowledge of module 6.1.1 cellular control. Okay, so Chidima, you're saying that you want to eliminate the top 2 because they're modes of transport, bulk transport. So here we have both of these, you're absolutely right, are to do with bulk transport. You have your exocytosis, which is about exit. Endocytosis is about entering, so you are engulfing. If you're doing endocytosis, it's exocytosis, you are releasing it out to the environment, whatever that environment may be. So then we need to look at, right, okay, so we're talking about body plan of an organism. Well, we know that organisms are multicellular. So then the question is, if we know that the organisms are multicellular, Jokeman, thank you for that. You're saying Hox genes, so mitosis. So we're talking about, it can't be meiosis, because meiosis is only to do with making your gametes, the reproductive cells. And the reason we need the gametes is they're going to end up being haploid, because meiosis is a reduction division. So therefore, when you are making more cells, absolutely, mitosis results in cell growth. So if we're talking about that overall body plan, we need to have more cells being made. Good, well done, guys. And if you are on TikTok and you want to see the questions, please do jump over to my YouTube channel which is Biology with Christine. We are doing paper 2021, and it's paper 2. Okay, so let's keep going with this question then. So the next one that we are looking at is genes are not expressed during cell division. So here we have the genes are not expressed during cell division because chromosomes are more tightly wound around histone proteins. So I love multiple choice questions because a lot of the time they're giving you the definitions that they want you to then give back to them when it comes to the open questions for you to answer. So I would suggest if you are doing the multiple choice questions correctly and you're taking the time, I would be rewording this, figuring out what my keywords are and helping myself make my own flashcards if I were you. But anyway, let's keep going. So genes are not expressed during cell division because chromosomes are more tightly wound around those histone proteins. Now, the fact that they've told us it's a histone proteins, I'm just going to add in a bit of note here. That straight away in my head, I would be thinking, well, they've told me it's histones. Anytime they mention histone proteins, we're talking about eukaryotes. My brain is really not with it right now. Guys, do bear with me today. Today has been a bit of a day and my brain isn't functioning quite as well, but it's all good. Okay, so which of the following shows the level at which gene expression is? So we're talking about gene expression is being controlled when DNA is more tightly wound during cell division. So if you have got your DNA, so we're going to wrap our DNA twice around our histone proteins. So what you should note is in eukaryotic organisms, we have got these histone proteins where our DNA is wrapped around those histone proteins, and that therefore, I'm not sure you're feeling the same thing that I'm feeling today. Well, I hope you're not, because I'd be very sad for you if I am.
I am.
Oh, really?
Yes.
Well, better now.
It's not been good. Mm.
It's not, it's been a day. My Nana unfortunately passed away about just over an hour ago. So it's yeah, it's a sad day.
And you're alive.
I am because I want to ensure that I help you guys. And also I've been preparing for it over the weekend. So she's now not in pain. So that's obviously a very good thing. Anyway, let's move on. Keep yourself focused. So we have our DNA, which is wrapped around our histone proteins. And the fact that it's wrapped around those histone proteins means that we're not able to access it with enzymes. And we're talking about enzymes such as helicase, or because we're talking about gene expression, we can also bring in our RNA polymerase. So if we know our RNA polymerase and our helicase are not able, thank you, Iris, are not able to actually access the DNA strands, what does that tell us? Well, that tells us what's RNA polymerase for. RNA polymerase, please stop being nice because you might actually make me more thingy with it. We're all good. RNA polymerase is for transcription. So we have got then our options. So post-transcriptional. So post-transcriptional. Now, Chidima, I know you are year 12. Much better. However, this is coming up in year 13. Thank you for that.
Can you stop scaring me?
Post-transcriptional is talking about after transcription has happened. So this is where you have your exons and your introns. And what we're going to do is the introns will be spliced out. So if you're year 13 or year 14 repeating, this is an important thing for you to understand. If we're talking about post-translational or translational, we know that translational is where we're going to be producing our polypeptide chain at the ribosome. So if we're producing our polypeptide chain at the ribosome, we therefore are needing our mRNA. And just to add in here, it's mature mRNA. We'd also have our tRNA involved, and we also have the correct amino acids being brought. Now, if it is post-translational regulation, what we're talking about is a precursor protein. So the protein has already been produced, but what needs to happen is that protein is going to be modified in some way. So earlier we talked about, or Chidima, you talked about the fact that a glycoprotein wasn't, one of the answers wasn't correct because the glycoprotein is the one that acts as a receptor for cell recognition. So if we talk about a glycoprotein, so if you think about your glycoprotein, the glyco is where you have a carbohydrate chain. And that carbohydrate chain is attached to a protein. So what we've done is we've modified the protein by adding a carbohydrate chain. We have produced the protein through translation. We've then modified it after translation has occurred. Now, going back to this whole thing about the histone proteins have DNA wrapped around it really tightly, especially if we're talking about prophase during the mitotic division. So prophase 1 is where your chromosomes are going to condense. If those chromosomes are condensing down, well, Saja, what year? This is 2021. You jump over to my YouTube channel, you actually see the question that I'm talking about and the notes I'm writing. Also, it means that it's available for you to view later on if you want to. Unfortunately, there's a lot of messing around on TikTok, which meant that I'm kind of trying to do both at the same time. So yeah, if you're just joining me on TikTok, I am over on YouTube, and I've actually got in the link a Zoom session that you can join. Back to what I was talking about. So the histone proteins, the DNA is wrapped around it. During prophase 1, you know that you've got your condensing of the chromosomes. During prophase, when we're talking about that in mitosis as well, you've got that condensing of the chromosomes. So to supercoil, which is what we call the condensing, to supercoil the chromosomes into the visible part of it, that is where we are going to post-translational modify the histone protein. So this is I'm going to give you a bit more information that you need to know. In the exam, they may ask about this, but they'll give you that information. So if they talk about acetylation, or if they talk about methylation, they're talking about you adding, even phosphorylation, adding an extra part, whether it's an acetyl group, whether it's a methyl group, whether it is a phosphate. By adding an extra bit on, you're modifying that protein. Therefore, that is a post-translational modification. So it's important whenever you're given these types of questions that you do take the time, but also by doing it this way, what we're doing is we're actually revising all the topic area, not just answering the question. And so many of you do that. You'll sit and you'll go through the question and you'll look at the mark scheme and you will have just ticked, yep, I got it right, happy as Larry, moving on. But then what they do is they take a very similar question, they just switch around slightly, similar wording, but you get a little bit lost because it looks so similar to one that you've seen before. Okay, let's move on to this next one then. So the next question is where we're looking at antibodies. So antibodies form part, they love a protein question. And this is one of the things that I would say where you have to note how proteins come through the whole topic area. So antibodies form part of the immune response. Which of the following types of antibodies bind to the antigens on the surface of the pathogen? Now, in the exam, there will always be little hints in the question. So many students will just read the question, skim read it, then jump ahead and go, oh, yep, all three of those. are types of antibodies, but then they've missed what the question actually has asked. So the question specifically asked, hi Maria, specifically asked about which antigens are going to be on the surface of the pathogens. So opsonins, what are they? The opsonins, remember, are about tagging and making the pathogen visible, i.e. more recognizable to our phagocytes. Agglutinins, what do? They clump the pathogens together. So therefore, because they're going to clump the pathogens together, that is going to be where they're going to stick to the antigens. Now, I'm using very lazy terminology right now. And this is something I want to highlight with you. A lot of you do the same thing in your answer. You will use lazy terminology. And what I mean is using words like they're going to stick to the antigen. No, they're not. They've given you the answers that they want you to use in the question. So again, I said to you, use multiple choice questions for figuring out how you can make the right wording. So you always want to use the term they bind to the antigens. So how is it that they're able to bind? Because they have an antigen binding site. So it's really important whenever you're doing your revision that you are using correct terminology, as well as when you're trying to describe to someone else or yourself. So always try and make sure that you pick up on that wording that's being used in the question so that you get into habit of using the correct wording, because honestly, it will make a big difference. The more you practice using it, the more likely you will write it down and not use the wrong terminology. So let's finish this question off then with regards to what it's saying. So you have your antitoxins. So what's going to happen? Well, these antitoxins are going to bind to our toxins. So they're going to bind to our toxins. And because they're going to bind to our toxins, where are our toxins? Well, the toxins are the ones which are the chemicals which have been released. So these chemicals have been released by the pathogens. Hi, Paul. But they're not actually on the surface of the pathogen. And because we know they're not on the surface, we can remove number 3 from our answer. So therefore, we've got number 3 being removed. And because number 3 has been removed, we can get rid of A and C. And therefore, we're leaving the only correct answer, which would be, because we've said 2 is also correct, it cannot be D, therefore it leaves us with B being the only right answer. Okay, so let's move on. Right, question 6 then. So for those of you who are just joining, we are doing 2021 paper 2, and we are doing question 6. So primary immune response. Again, look at our definitions that they've given to us. So the primary immune response involves differentiation and clonal expansion of certain white blood cells. So they're giving us some key words here that they would want us to then give back to them if we're going to give any definitions. So which of the following cells results from the clonal expansion? So clonal expansion, remember, we're talking about clones. Clones are genetically identical cells. Those clones are produced through mitosis. Oh my word. Let's try and do that again. Mitosis. So because we know that we're going to end up with genetically identical cells, and those genetically identical cells are being produced through mitotic division, mitosis, we are expanding the number of cells that we have. So if we understand that, which of these are during the primary immune response? So there's two different ones. There's the primary, oh, 1 second, there we go. Primary, which means it's the first one. And then you have your secondary, which is the second. So they're asking about the primary immune response because they're asking about the primary. Why can I not talk tonight? Let's try that again. Because they're talking about the primary immune response. They want to know which of these are going to come from that selection, the expansion, the mitotic division. So they are differentiated. How are they going to differentiate? Well, our B cells, if I've got a B cell here, when that goes through mitotic division, that can go through mitotic division to produce my plasma cell and also my memory cell. So that is where we're going to have. No, it's not that. Paul, you missed it when I said, unfortunately, my nana died at 7:15 today, so I'm feeling a little bit not quite with the sessions as I would be normally. But celebration for anyone who is Irish, because yes, you are correct. It is actually, I forgot that, it's St. Patrick's Day today. So well done.
Thank you. I am Irish.
How are you?
No.
You could be. I wouldn't know. So if we look at the T cell and we think about what could the T cell potentially go through with regards to production. So you need to know about your T helper cell. You need to know about your T killer cell. You need to know about your T memory cell. And I was about to add in your T effector cell, but they've removed that. So let's just take that one away. So therefore, would you get through the process of clonal expansion your T memory cell? Yes, you would. So therefore, that tells us that for this answer, we could have all three, which would therefore be A. Thank you. Thank you, Lynn. Okay, moving on to the next question then. So if we look at lupus then, so we're being told lupus is an autoimmune disease. So again, make sure that you're highlighting what it is that they tell you. Now, lupus has actually been removed from the specification for the 2025. And when I say it's been removed, it doesn't mean they're not going to ask questions about it. They've just removed it from the list that they want you to know. You used to have to know 3: lupus, diabetes, and rheumatoid arthritis. Now they've just put in arthritis. They've made it easier for you. So they've removed the other 2, but that doesn't mean they're not going to ask a question on it, because if they are going to ask a question on it, it will be about what is an autoimmune disease. So in this one, we are being told that it is an autoimmune disease. So auto means it's going to kill our self cells. So it doesn't recognize our own antigens as being self antigens. It actually thinks that there's some form of pathogen, some form of problem that it needs to eradicate. And so therefore, if we have an autoimmune disease, we are going to be killing our own cells. So in this case, specific to this example that they've given us, this affects cells in the skin, your joints, and internal organs. So these are very specific places that are affected by the immune system, because the immune system does not recognize these cells as being self cells. Now, if they're talking about rheumatoid arthritis, again, to give you information, use that to guide you on what they want you to answer. So, which of the following is likely to be an effective treatment for lupus? And remember what I've said, it's the why are the wrong answers wrong, not what's the right answer. That's not going to help you. So, immunosuppressant drugs. Is this going to be a good treatment for lupus? Well, if we are going to suppress through the use of drugs the immune system, we're therefore going to stop the immune system attacking cells. So that seems quite sensible that that would be the right answer. But if it is the right answer, then why are the wrong answers wrong? So that would be where we want to go with this next one. So injections of antibodies from someone who does not suffer from lupus. So if we're going to inject antibodies, what are they for? They're for fighting pathogens by binding to the antigens. So if they're going to bind to antigens, is it going to be effective in injecting these antibodies? Well, no, because we know lupus is actually a disease that's affecting our skin, joints, internal organs. It's not caused by a pathogen. Somatic cell gene therapy. So again, what does that even mean? So somatic cell gene therapy, we're talking about our body cells. So we have our body cells and we're going to do gene therapy. And what we're going to do is insert a gene, which therefore means that there's going to be proteins produced. Well, again, is this going to be a good treatment for lupus? Well, no, because remember, lupus is an autoimmune disease where we have our cells are not recognized and our immune system is going to fight against it. No, they don't. They don't. Thankfully, there's so much content already that no, they don't have to know it that full. And then vaccinations, going into this one, vaccinations, what you're going to do is you're going to potentially inject the antigen, and this should stimulate your immune response. Well, we're not wanting to stimulate an immune response. Instead, we want to stop the immune response. We want to suppress it. And that's why it therefore means that A is the right answer. Now, I did say the vaccinations with injecting the antigen, or it could be a weakened version. But be aware, you don't just get an injection. You can, if, for example, you're a child, my daughters actually got a nasal spray. And that vaccination of the nasal spray was where, again, they put the weakened version of the pathogen into the body so that they are then having an active immune response. Their lymphocytes are going through clonal selection and clonal expansion. Therefore, you're getting more of them. Okay, so let's keep going with these questions then. So we're still talking about lupus, but this time, this question now is looking at your maths skills. So it is looking at your ability and the math skills that you have. So you have the, oh, 1 second. Sorry if you have been waiting a while. Let's let Andreas in. Okay, so lupus is really a rare condition affecting approximately 0.025% of the population. However, 9 out of 10 sufferers are women. The population in 2019 was approximately 64 million. So, which of the following estimates shows the number of men in England and Wales suffering from lupus? So what would we do for this one? So we've got 0.025 of the population that are going to have lupus. So if you have 64 million in the population and you've got 0.025% of them will have lupus, that would be the first thing that you want to do. You want to work those numbers out, and I can't, where's my calculator? 1 second. Because I'm using my phone here. I'm so used to grabbing my phone. Let's go for this. Yes. Um, and then what you need to do is once you've got your 64 million, you've got 0.025, you then know 1 out of the 10 is going to be men. So 1 out of the 10 would be men, or that would give you the correct answer, which is 16,000. So that is a case of you using the numbers that they've given you and then working that one out to give you the correct answer, eliminating the others. I'm glad. Takes me a while to even get the calculator up. Sometimes I'm like, how do I find that again? And it's one of those things I would say, yes. So you want to find the 0 point, because you're told only 0.025% of the population have lupus. So because you're only, yeah, my phone's up there because it's the one that's doing the TikTok, and then on here I've got my computer. So I'm so used to doing this. But I would say to most students, make sure that you actually have practice using your calculator, because the more you practice using your calculator, the more you're going to actually feel confident, comfortable in the exam. So you want to ensure you've got, yep, well done. So multiply the 64 million by the 0.025, and then you're told 9 out of 10 are women. So therefore, you only have 1 out of 10, which are your men. And so that's why you want to bring in that part second. So does that make sense? Good. Perfect. Okay. So which of the following substances is not required? Why would you do 1 minus the 0.025? The 0.025 is about the population size, how much of that population size. So you need to work that figure out first, and then go from there. The fact that only 1 out of the 10 are actually going to be men. Okay, so back to this. Which of the following substances is not required? And this is important, not required. So DNA sequencing then, what is DNA sequencing? Well, DNA sequencing is where you're going to take a small sample of the DNA and you are going to sequence, so you know, does it go A, T, C, G, G, A, T, C, A, G, C, A, T, C, A? Or does it go A, T, C, C, G, A, T, T, C, C, A, T, C, G, A? So DNA sequencing is that you are going to sequence, you are working out what the DNA sequence, those nitrogenous bases, what order do they come in? Because that order is important, because that determines our amino acid sequence, and that amino acid sequence therefore is going to determine our polypeptide chains primary structure and the way that it's going to fold and interact. So if we're going to sequence it, we need to take a small sample of the DNA, and then we're going to need to amplify it and then put it through a sequencing process. So it tells us some key words. So we need DNA polymerase for what? Well, DNA polymerase is needed for joining adjacent DNA nucleotides. Well, if we're going to amplify, we are going to need to do DNA replication. So therefore, DNA replication is required. So we're going to need DNA polymerase. So this is asked which of the following is not required. So we're going to remove our DNA polymerase. Yeah, I will actually. Chidima, I will do AS papers. I think I'll do that on Wednesday because I have done quite a lot of A-level papers, but I'm very aware that the A-level is literally in 10 weeks. For those of you who are AS, you've got a whole other year of me to go through this and to learn this content. But for those who are actually in year 13, this is it for them. So your mocks are in a month and you're in a very good position for those mocks. Just, you're okay. You get the scores that you need. If your teachers are not actually seeing that already, then that says more about them because you're already showing that. I bet you can't, Lynn. Okay. Now, this is an interesting one because if you haven't actually done DNA sequencing, okay. So Andreas, you will cover this. And what I suggest is have a look at my videos, which are on this topic area, which is, so we're doing DNA sequencing, which is manipulating genomes, 6.1.3 in the playlist. So RNA nucleotides. Now, this is where, Paul, you will, so for those of you who don't know who Paul is a retired professor. You'll know this as well. It actually annoys me sometimes where they add in bits and say it's wrong, when actually, if you think about the fact that your primers, your RNA primers are actually nucleotides that are made up of RNA. So they're very sneaky with the wording that they use in these questions, because if you have a primer, the primer that is going to be used is actually an RNA primer, and then that RNA primer gets removed. So for those of you doing the actual exam, yes, you need a primer. A primer is needed because DNA polymerase cannot add on nucleotides without a primer. So this annoys me that they teach you DNA replication without actually teaching you about primers. So yes, you can get DNA primers, but most of the ones that are used in the labs are the RNA primers as well as DNA primers. Exactly. But it's that whole thing of they teach some things and not others. So they've just used the term primer, which is required for DNA sequencing. So therefore, that is not correct. A terminator base is one that is needed to terminate the strand. And you will learn about that in the fact that the terminator strand or terminator base is going to terminate the strand at the base, and it's going to have a specific dye attached to it. And that dye is necessary for identifying that base. So whether it's adenine, whether it's cytosine, whether it's guanine, whether it is thymine, each of them will have a different color dye associated with it. And you'll learn about those different ones. So C was the right answer. Yeah, don't worry about the primer stuff. All you need to know is that it needs a primer. So don't worry about it. Sorry, that was me having a little conversation with Paul with regards to where they use the term in there, but they don't actually give you the information. So many students who go on and study biology at university start their lectures with the lecturer saying, what you learned in A level wasn't actually true. Now we'll teach you the right stuff because they don't think you can handle the information. Okay, so let's move on then on this one. Question 9 is from 6.1.3. So it is manipulating genomes. Right, DNA fragments can be separated using gel electrophoresis. So we're going to use gel electrophoresis. We're going to separate fragments of DNA. So which of the following explains how gel electrophoresis is able to separate the DNA fragments? Well, the first thing I would say to note is our DNA, we should know, is negatively charged. So because we know it's negatively charged, that therefore tells us that that negative charge is going to be repelling from a negative electrode and moving towards a positive electrode. So that's the first thing I would say. You should know that DNA is negatively charged, and then it expects you to go from there. So what can we say? Well, DNA carries a negative charge. Absolutely. So we can now remove our C and D. So now we're down to a 50-50 shot as to which one to eliminate, leaving us with the only right answer. So let's look at A. DNA carries a negative charge and large fragments are pulled more strongly than small fragments towards the positive electrode. So if they're going to be pulled more strongly towards the positive electrode, that would make you think that there's a difference in the ability to pull. So that doesn't sound right in this question. The fact that they're being pulled because the electrodes are a negative and a positive electrode. And why would a larger fragment have a stronger pull than a smaller fragment towards that positive electrode? So let's look at what B is. DNA carries a negative charge and small fragments are able to travel more quickly than large fragments towards the positive electrode. So what they've done with this question is they've given you something that sounds almost like it could be true, but actually you should know that it's not true. You should know that it is the small fragments that are going to travel more quickly through the gel, the agarose gel that you are going to be using. Okay, let's move on to 11 then. I'll probably just do the multiple choice questions and then finish for tonight, guys. Okay, so gene sequencing has a number of uses. Which of the following, again, they love this not. One of the things that I do notice is sometimes in the multiple choice, they will put a lot of not questions if in previous years they've done a lot of assessing you on sole topic areas. So the not questions actually get you a bit confused because what they want you to identify is things that shouldn't be happening. So in this case, we need to look at again which ones are correct this time so we can eliminate the correct answers. So which of the following is not a use of gene sequencing? So if we're going to sequence a gene, well, what is a gene? A gene is a section of DNA. It's a section of DNA that is going to code for the amino acid sequence in our polypeptide chain. So then we look at the next part and say, okay, so if we're looking at this, is it going to determine the amino acid sequence of a polypeptide? Well, if we're going to sequence the gene, we're therefore going to sequence, remember, whether it is AAT, CCGA, TA, just making these up on the spot. If we're going to sequence that, we're therefore going to know what the codon is. And if we know what the codon is, we'll know which amino acid is going to go in that sequence. So therefore, if it is methionine, then glycine, then alanine, we will know from doing the gene sequencing. So therefore, determining the amino acid sequence is a correct statement, therefore is not the right answer to this question. So then we'll look at the next one. The classification of newly discovered organisms. Well, if we're going to sequence the gene, and we're sequencing the gene, we can then compare that to others and therefore use that in an analysis of whether or not they are classified as being the same species or a new species. So again, this is a correct statement, so therefore removing it from being the answer to the question. The next one, which of the following is not a use of gene sequencing? The polymerase chain reaction. Well, polymerase chain reaction, PCR, is about amplifying minute samples of DNA. So you can then use them. So would this be a use of gene sequencing? No, because PCR is about amplifying, but that therefore means if C is correct, D must be wrong. So can we eliminate D? So the selection of the correct vaccine in the disease outbreak. Well, if we're going to select the correct vaccine, we're looking at the potential antigen so that we can therefore produce the correct, oh my word, let's try and combine that together, correct antibody, because we want to make a antigen antibody complex. So therefore, knowing the gene, the sequence of that gene that codes again for that amino acid sequence that therefore is what forms our antigen, means that that would be a use of gene sequencing, therefore leaving us with the only right answer, which was C. Oh, okay, I'm gonna have to zoom out on this one. Okay, so here we have a question about a plant that is a wild type and a dwarf variety. So they investigated the growth of the plant. Now, another thing I would do is I would say, right, well, they've told us Brassica rapa, so they've told us the genus and they've told us the species. They've given us the two names. They've given it in a capital letter and a lowercase here, and they've written it in both formats. They've written it as a full name. They've also abbreviated it down to a capital B dot rapa. So just to note, they can do it in any which way with regards to the way they display it. So just note, they could ask you about how you would approve. That's great. Biology is the best subject. If you do taxes, then LZ, I'd say you're AQA because we don't do it in OCR. Taxes is just talking about whether it's chemotaxis or phototaxis, you're talking about movement due to the stimuli, whatever that stimuli is. So, okay, back to this question. So seeds of both varieties were planted and the heights of the stem were measured 20 days after. So again, one of the things I would say to note is whenever you see questions like this, they are giving you little bits of information because the questions in the paper have to be good questions which give you accurate evidence of the skills they're going to either be assessing or the skills that they want to test whether you know how to improve on. And what I mean by that is, do you know that your table of results, your independent variable should be on the left side of the table, and then you would have your dependent variable on the right side, and you'd also have then process data at the far right hand side. You all right?
I'm just too funny. I'm just too funny.
You are. Oh, I just saw that because I'm still doing the stuff. I'm seeing the comments a bit later. Okay, back to this then. Where were we? Process data on the right-hand side. And then what we're looking at is other thing to note in your table of results, second, is they were all given to 1 decimal point. So therefore, you need to make sure if you ever get any which go to a whole number that you don't forget to put in that 8.0 or 92.0. So the data they give you in these multiple choice questions needs to be accurate unless they're assessing you on can you identify the problems that are there. Okay, so moving on, which the following correctly explains the student's results? So now they're asking about variation in height between varieties. So we highlight what's the difference here. So between variety, or is it within varieties? So that's the first thing that you need to be able to eliminate. Which one of these is it between varieties or within? So if we go back up, we're told that Brassica rapa is a plant that occurs in wild type. Yes, so it should always be your independent on the left-hand side, then your dependent variable, and then the processed data, if you're going to do processed data, will always be at the far right-hand side. So it could just be your raw data. So you've got your independent variable and then the raw data with no processed data. So that'd be left, independent variable. And the way I used to teach it as well when I was doing this with GCSE is I'd always say my independent variable is on my left-hand side and on the bottom of my graph. So table, graph was how I used to remember it. Left-hand side for your independent variable, down on your y-axis when you're talking about your graph. So using that as another guide. So if you were going to graph this data out, you would therefore have your wild type and dwarf at the bottom, and you would also want to have this as a bar chart and not as a line graph. So what we have then is a plant that occurs in a wild type and a dwarf variety. And so therefore we're being told that they're not the same. So we are not within varieties, we're between the varieties. So that's the first thing we can do is we can eliminate the C and D, that they're not the right answers, leaving us down to that 50% chance of being right. So now what we need to do is we now need to identify, well, what's the difference between the rest of the question? So A, the variety in height between the varieties, the variation even in height between the varieties is continuous and polygenic. That basically means poly means many genes. Continuous is where you have any value, i.e. intermediates. And then if we're looking at the other side of it, we're looking at discontinuous and controlled by 1 or 2 genes. So in this case, we're looking at it being discontinuous. So if it's discontinuous, we would therefore graph this out as a bar chart. And we're therefore saying that it's controlled by only one or two genes because they are separate, no intermediates. So in that case, what you can have is you can look at this and see that which of these have we got? Well, we've got a wild type and we've got the dwarf, and we're looking at the means, and therefore that would be that this is actually variation in height between varieties, and because it's between varieties, it's controlled by one or two genes for this one. So that would be what they're looking for. Okay, moving on. Question 13. So if we're looking at the graph, then with this one, we would actually draw this as a bar chart. So you would have your wild type, and then you'd have your dwarf, and then you'd have your, oh, that's really bad way around if I were to actually sketch it properly. The wild type would be much higher, and then you'd have your dwarf. And if you're going to do a statistical test against that, you would therefore be doing a t-test. My brain really is not on it today. Try that again. T-test. So you would have your variety at the bottom, and then up here would be your mean height after 20 days. And that would also be in centimeters up here. And it would be a t-test because you'd be comparing that. Because with continuous data, for it to be polygenic, you would have all the intermediates. So you would have every number in between, whereas we've got two very different values here. And you can see that quite clearly when we look at the data being drawn. And so therefore, it's not going to be many genes. If we look at, for example, skin color is to do with the pigmentation that's put into your skin. Well, it's not a case of that you are white or black. You're every shade in between. And because you can be every shade in between, it therefore means that there are many genes interacting in producing the color that you have in your skin. And so therefore, it's that whole thing of when we talk about continuous data and continuous variation, that normally means that there's any numerical value in between or any value in between intermediates, and it's many genes interacting. So I could do it on petals. For example, if we had red and white, depending on how many genes were interacting, would result in lots of different shades. You wouldn't just get red or white, you'd have many different shades. So I hope that helps. So if you see a set of data and what you're looking at, for example, is normal distribution, which would give you the bell curve. So if this was continuous data, we should be able to get what's known as that normal distribution, where you'll get your bell curve, where you would have the majority of your values in the middle with few at either end. So when you look at, for example, the variability in your data, what you're looking at is that standard deviation plus or minus 1 standard deviation is always going to be 68% of your results. And that would give you what's known as that normal distribution, that the majority of them are found within the middle. So if you were to take everybody in your class and line them up in high order, you would literally have all the way from the whoever the shortest person is, right the way up to whoever the tallest person is. But you'd find a majority where you're like, oh, maybe you guys need to just move around slightly. Oh, wait, you're there, because they'll be very close, but there'll be differences. So you've got every possible intermediate value. So yeah, I hope that helps. I'm still considering on that one. I'm still figuring out what's going on with regards to the course, so I will let you know on that one. Mohammed, if you're looking at partial pressure, you want to look at transporting animals, go to my YouTube channel and look at transporting animals. No, yes, no, exchange, hold on, partial pressure, bore shift. Yeah, you want to go to transporting animals and you want to look at my bore shift dissociation curve video on that one. Okay, so let's move on with this one. Moving north from a large solitary tree in the school field, some students studied changes in plant species. They laid a tape measure, right? Again, so here we have, they are sampling because they are studying. So I'd be writing a note to myself that says this is a sampling question. I wouldn't be surprised if a sampling question does come in multiple choice this year because the sampling question was a chi-squared question last year. So they laid a tape measure due north from the base of the tree trunk and dropped a quadrat at 1 meter intervals for up to 15 meters. So again, if you're making revision notes, what I would suggest you do is use this as a guide. They're telling you how to actually do this when it comes to answering the question. If they were to flip it and ask you to describe how you would do it. So what are we doing? We're taking a tape measure. We're placing our quadrat down every 1 meter. And we are then sampling in that quadrat which of the species that we're looking at. So is it arbitrary? What does that mean? What does that actually mean if it's arbitrary? What does it mean if it's random? Well, that's the question I have. What do they mean when we talk about something being an arbitrary unit? What do we mean with that? So if we're saying it's an arbitrary sampling method, what does that mean? What they love to do is throw in some of these words that you've heard before and just to try and confuse you. Yeah. So an arbitrary is just a standard. So when we talk about arbitrary units, we're talking about standard units. So therefore, arbitrary sampling method, is that one? No, it's absolutely not. So what is random sampling? Well, random sampling would be where you take 2 tape measures. And then what you would then do is you would lay those tape measures down until from 0, let's say, to 30 meters, and you would get random coordinates using a random number generator. So that would be a random sampling technique, which is used using random numbers generator or some form of way of getting the random coordinates. Stratified, when you do it stratified, what you're talking about is if I have a sampling area, but it's made up of different size. So this may be a bog, this may be a heath, and this may be farmland. What I would then do is I would look at, well, what have we got? We have got, this one is an area of 1,200 meters. This one is 800. This one is 400. So therefore, what should I do? If I'm going to do stratified sampling, I would do proportional samples based on the area. So therefore, they're not doing that. It's not stratified, and you're absolutely right. It is systematic. Okay, last 2, and then I'm going to stop for tonight. So which of the following best defines the term species richness? So species richness is one of the definitions that you should know, especially if we're looking at species biodiversity. So if you're going to do Simpson's index of diversity, you're going to be looking at whether or not this one is. Chidima, this one is module 4. This is biodiversity.
I don't know how to do it.
So you should have done Simpson's Index as part of the biodiversity topic, which is module 4.
I don't think I've done that yet. We've not finished
No,
AF completely.
if you're doing communicable diseases or classification evolution.
Classification evolution, and then with my other teacher, after we finish transporting animals, we'll do communicable diseases.
That's why you haven't yet done biodiversity, which is the other part of module 4.
Okay.
So in this one, what we're looking at is the richness. So if I have a site called A and a site called B, and if I'm going to sample those sites and I have dandelions, I've got daisies, I've got buttercup, and I have got thistles. So if I have these different plant species that I'm identifying, and I'm just making this up. So I've got 4, I've got 7, I've got 9, I've got 0. On this one, I have got 2, 4, 9, and I've got 25. Okay, so if I'm looking at the species richness, looking at site A and site B, I have a greater richness in site B than I do in site A. So therefore, what am I looking at? I'm looking at how many species have I got? Well, in site A, I've got 3 species because there was none of the thistle, whereas in site B, I've got 4. So therefore, the number of species in an area is the richness. So therefore, the correct answer was B. But let's just confirm that the other answers are incorrect so that we can guarantee that we know our topic area. So the distribution of species over an area. So the distribution of species over an area is just showing us the distribution of species over an area. That's not the species richness, that's where they're located. The relative abundance of each species in an area, or the relative number. Now, this is the bit that they love to throw in there to try and confuse you. The relative abundance of species in an area is the, right, richness again. It's not what I meant. The evenness. So if you look at my numbers, what you can see is I have got 4, 7, and 9. They're pretty evenly distributed in species site A. When you look at site B, you've got 2, 4, 9, and 25. Therefore, there is not an evenness in site B compared to site A. So it's important when you're looking at evenness that you know the difference between whether it's species evenness or whether it is species richness. So give me 1 second. It's going to add in. So if I just switch screens a second, switch back to this one. So when you're looking at the population and the species, know your definitions, but also know the definitions for species richness and species evenness. So species richness, the number of different species in the area. So here we can see I've got A, B, and C. We can say that species area A has the lowest species richness because of there being no millipedes, so there's only 3, whereas B and C both have 4 each. And species evenness is that relative abundance, and that means the number of individuals. So just be sure when you're doing this and you are answering the questions that you know the difference between the two. Because what they'll try and do is they will try and confuse you by giving you a question which is almost right. Okay? And so therefore, the relative abundance of each species in an area, the relative number of individuals of a species in an area. So do make sure that when you're going through this, that you are eliminating the wrong answers, but also using it to guide you on your definitions. Okay, so last one that we'll do tonight then. Biodiversity can be considered at different levels. Remember, there are 3 levels. So we've got 3 levels. We've got the habitat, we've got the species, and we have the genetic level. So an area of woodland habitat has a high Simpson's index of diversity. So a high Simpson's index, so the Simpson's index goes between 0 to 1. The higher it is, so 0.99, for example, the more diverse it is. The closer it is to 0, the less diverse it is. So which of the following describes an area with a high Simpson's index of diversity? So we're looking for Simpson's index. Now, is Simpson's index about genetic, species, or habitat? So therefore, they're looking at, well, you're told it was a woodland habitat. So what we can say is we can automatically say it's not going to be a high habitat one because they told us it was only a woodland habitat. We can say that it's not genetic because it's not giving us any information about the alleles. So we know it's not to do with the genetic biodiversity, which is the variety of alleles within the species. So therefore, the area is high in all of the levels, absolutely not in all of the levels, leaving us with the only right answer, which is C. So it mentioned woodland habitat, but to have a high Simpson's index of diversity, you're bringing in species richness and species evenness. So if you're in a area of a woodland habitat, it's a woodland habitat. If there's a high Simpson's index of diversity, you'd be looking at a high habitat biodiversity would be lots of different habitats within one area, whereas you're told it's a woodland habitat. So it can't be a high habitat biodiversity because you're told it's just a woodland. They really are. And that's one of the things as to why I would say using the multiple choice practice questions as a way of revision is a good thing. What's your opinion on STEM up? I don't know. I haven't seen them. I can't tell you if they're any good. I would say if you find them useful, great. Sometimes students can become a bit too reliant on someone else's flashcard. And if you're not actively doing some form of assessment on yourself and how you're using them, then you are passively trying to get the information. So it really depends what type of learner you are. I can't say because I don't know what they're like. And actually, someone spent the time making them, so I'm sure they'll have notes in there, and I'm sure that they will. Yeah, absolutely, Paul. I'm sure that they will have some good information, but I don't know whether they're really good to suggest using them or not. Sorry. Guys, thank you so much for joining me again tonight on tonight's live. And I will be coming back on Wednesday. Thank you. You're very welcome. Thank you. Thanks, guys. And yeah, keep watching my videos, keep sharing, keep liking, and If you are interested in my Easter course, please do get in contact. If you haven't seen, I've just uploaded my workbook on module 1, so do obviously check that one out as well. But thanks, guys. Have a good evening.